So this tutorial is to help walk you through a comparison of DNA replication versus transcription versus translation. I think it's helpful for students to study these processes side by side so they can clearly see the similarities but also clearly see the differences, because each of these processes is really for a different purpose, and so you're going to see a lot of differences. So what I would suggest is to go ahead and print out this sheet, and then as I walk through this you can write the answers in, and then you have kind of a little study sheet. This is intended to help you with some, I would say, confusion that typically happens on the day of the test if you haven't intentionally studied these three processes in sort of a side-by-side way. So the first, we're going to start with DNA replication, and I'm going to go through this whole column. And you'll remember that DNA replication copies DNA. So we're copying from a double-stranded DNA template. And I would also say, you could also say that the template is, whole chromosomes, because during DNA replication we're trying to, replicate entire chromosomes. So monad chromosomes after replication become dyad chromosomes. And so DNA replication is typically a very global, I guess you could say, process inside the nucleus, meaning it affects the whole genome. And it's both strands of the DNA are copied. The initiation point for DNA replication is called the origin of replication. On a bacterial chromosome, there's one origin, and on a eukaryotic chromosome, there can be many, many origins of replication. Directionality, students will often immediately say 5' to 3' is the directionality, and that's true. So on each strand, the new strand is built from 5' to 3'. You could also say that from the origin of replication, that DNA replication occurs bidirectionally. All right, so that would be another concept of directionality, because you have two replication forks, one from each replication bubble. You have one fork that moves, to the right, and one fork that moves to the left, and so that's bidirectional replication. Now, the critical enzymes, there's a lot of them. Remember that there, and I would include proteins in this as well. There's the initiator proteins. Initiator. There is then helicase, and then there's the SSBPs, single-stranded binding proteins. There's the gyrase, sometimes called topoisomerase. That's the enzyme that reduces the overwinding or torque ahead of the replication fork. And then when you get into the actual synthesis of the new strand, you have primase, which builds the primer. Then you have DNA polymerase 3. That's the one that does the main part of the synthesis. Then you have DNA polymerase 1. And that's the one that removes the primers and replaces them with DNA nucleotides. And then you have the ligase. So you have this laundry list of enzymes that are needed in DNA replication. DNA replication happens in the nucleus because DNA always stays in the nucleus. Now that would be in a eukaryotic cell. In a prokaryotic cell, DNA replication happens at the nucleoid region. Temporal location, meaning timing. When does DNA replication happen? In a eukaryote, it happens during S phase. Termination details. When and why does DNA replication stop? Well, I think what you could say is that whenever you run out of template. meaning whenever the replication forks run into each other or run off the edge of a linear chromosome. So when all chromosomes are replicated. That's why it stops. There's just no more work to be done. And what's your final product? Replicated chromosomes. Or you could say dyad chromosomes if you're talking about eukaryote. Or you could say sister, chromatids. Those are all concepts of the same thing. Okay, so that's DNA replication. It's the way that the cell prepares for a cell division. It's the way that the cell makes copies of its genome so that it can pass a full copy of the genome to each of the two daughter cells. Now we move on to transcription. In some ways, transcription is going to seem similar, but the purpose of transcription is just fundamentally different. The type of template that's used in transcription is chromosomal DNA, but it's only going to be one strand that's going to be copied. One strand of a double-stranded DNA molecule, you could say. You could also say that the template is a gene. So we're not copying the whole chromosome, we're just copying one strand of one gene within the DNA. So it's a much smaller scope. The initiation point is called the promoter. The directionality does go from 5 to 3' on the new strand on the RNA. But if we're thinking comparatively with what we wrote with DNA replication, you could say it's unidirectional, meaning it's only on one strand and it only goes in one direction. Now, what are the enzymes? There is simply RNA polymerase. Now, the RNA polymerase does have a subunit that functions as the helicase. So I'm going to say with a built-in helicase. But it's considered one enzyme. So you don't have this long list of enzymes, you just have one enzyme that's doing it. The cellular location of transcription, that is the same as DNA replication. It's going to be the nucleus for a eukaryote. For a prokaryote, it would be, at the nucleoid region. Temporal location, this is a little harder. Transcription is the way that the first step of gene expression, and so it can happen at all phases of the cell cycle, but mostly I would say you have more transcription during your gap phases, G1 and G2. But you can have transcription at any time. There will be low levels of transcription for some genes, even in other phases, but mostly G1 and G2. Termination details, why does transcription stop? It's because you reach the end of the gene. It's when RNA polymerase reaches the end of the gene, which we call the terminator. So we're not going to copy the whole chromosome to the end of the chromosome. We're going to copy one gene from the promoter to the terminator. That's it. And the final product is RNA. Now, students always want to say mRNA. Well, there's a lot of other types of RNAs, and they're made also by transcription. So RNAs, all types, I'll say. All right. So it's a different scope. It's a different product. There's a few things that are similar, but there's actually a lot more things that are different. And then translation. Well, translation is quite a bit different. I think the only reason it's confusing to students is that the word is similar. You have transcription and translation. But the whole process is quite a bit different. The type of template, if we were going to use the word template, I guess I would say an mRNA is the template for translation, meaning mRNA has the information that's going to be used to make a protein. The name of the initiation point, the translation start point, is called the start codon, and it's in the mRNA. And I should mention the promoter is in the DNA, and the origin of replication is in the DNA. So the directionality, the mRNA will be read, boy, having some issues. mRNA is read from 5 to 3. And the protein is built from N to C. Now N to C means free amino end to free carboxyl end. So the new amino acids are always added to the carboxyl end of the growing chain. The critical enzymes, there are enzymes that are built into the ribosome. So you have peptidyl transferase, but I don't really care if you know the name of that, but peptidyl transferase is an enzyme. And these enzymes are built in to the ribosome. So they're actually part of the ribosome. You also have enzymes called aminoacyl tRNA synthetases, and this is in the lecture, synthetases, so you have to know that one. tRNA synthetases, and those are the enzymes that charge the tRNAs. Charge means loads them with the correct amino acid. The cellular location, remember the ribosomes are in the cytosol. So you have the free ribosomes are in the cytosol. And remember, all translation starts on free ribosomes. That's part of the signal hypothesis. The ribosome may move from the cytosol and then attach to the rough ER, but all translation initially starts on free ribosomes. So the ribosome may become bound to the ER later. All right, what's the temporal location? It would be the same as what we had with transcription. You can have translation happening all the time, but you're going to have a big push during the gap phases. Termination details. Why does translation stop? It's because it reaches a stop codon, which is also in the mRNA. That signals the stop of translation, and the final product is a protein. So that gives you an overview of, or a little side-by-side comparison of these processes. You can see, well, it's really small. All right, thanks for watching.